Welcome to our campus at UMass Boston. It is constantly growing and changing itself like a developing embryo, if I may say. So today we want to take you on a journey to gastrulation and to explore some cellular movements that occur during development. And I want to start by asking this question: where do we all come from? Now, this may seem like a deep philosophical question for some, but as a biologist, I answer it pretty simply. We all come from a fertilized egg. So you see 3 eggs here, and try to guess which one of them will give rise to a sea urchin, which one of them will become a mouse, and which one will become a human. Well, without further ado, I will reveal my secrets here. And so it's fascinating to think how a ball of protoplasm and like a nondescript single cell can become complex, beautiful organism. This happens through the process of embryo development. And this is what I am most interested in. This is a complicated process. And to study development, we try to think what happens at the level of individual cells. What is going on as they're building these multicellular systems? We can distinguish 4 key processes that occur during development. Cell proliferation, when the cells increase in number. Cell specialization, the process when some cells become different from others in the embryo. Cell interactions, when you grow a big mass of cells and they have to do something together, they have to agree to carry out a common developmental event. And so to do this, they have to communicate. And finally, cell movements, when parts of the embryo move relative to the other parts of the future organism. Now, to understand our development is sometimes complicated. So we use simpler model systems to study that. And my favorite organism is shown here. This is the fruit fly, Drosophila melanogaster. And an amazing fact about fly development is how similar it can be in many aspects to the development of humans. We can detect all of these same processes: proliferation, specialization, signaling, and movement in the developing fly. And so, of most interest to my lab is the process of cell communication or cell signaling. Many years of research in this area, including some people who are present here in the audience, indicate to us that it's a complex process, and extracellular signals arrive at the cell and they initiate a cascade of events inside the cells that occur and are carried out through protein interaction networks inside cells to finally achieve an output, such as a change in cell shape, movement, or other events. And in my lab, we use a 2-pronged approach to study cell communication. First of all, we try to identify new interesting molecules in these signaling networks. And then we use our favorite organism, the fly, to try to understand the functional importance of these interactions. What do they do to carry out signaling and development? But today, let's focus on a very important event in the life of most organisms: gastrulation. Here, you can see a video of a gastrulating Drosophila embryo, looking at it from the ventral side. The head is here, the tail is here, and this is the belly of the embryo. You can see that a key event happening here is the movement of cells from the outside of the embryo inside. This is a key developmental event because it establishes the future body plan of the organism and starts the development of most organ systems in the future organism. And so today, my talk is divided into 3 parts. First, I want to peek with you into the internal workings of a single cell to see how can cells set up molecules to move things around. In the second part, we will take an even closer look at the function of these molecules, how they generate forces. And in the third part, we will pull it all together to study gastrulation itself. So the developmental processes involve the movements of multiple cells. But even in simpler organisms, such as this amoeba called Dictyostelium or a slime mold, you can see the precursors of very complex behaviors we can see in more complex multicellular organisms. You can see that this single cell is able to sense a source of an attractant that is coming out of this capillary and can reorganize its shape to move towards that source. And so I tell my students that you can play with a cell that you can, as if you're playing with your cat. All you need is a little laser pointer and it will follow where you show it to go. And so what is inside the cell? If we take this poor cell and fix it and subject it to electron microscopy, we will see something like this. The interior of the cell is packed with stuff. It is very dense, and it has inspired a lot of art, actually. You can see these beautiful illustrations by David Goodsell. They are very true to life. He is a scientist as well as an artist that represent the dense and complex environment inside the cell. But it is not hopelessly disorganized. To the contrary, we can find a lot of order and organization inside the cells. And this is because the interior of the cell is divided into these little systems that are called organelles. They're like little organs that perform certain cellular functions. And when my son, Artyom, got a school assignment to come up with an analogy for a cell, I think he found a fantastic one. He thought that the cell looks like a house. Look, there is a living room, the nucleus of the cell, of the family, right? Here we have a generator, which is analogous to mitochondria in the cell, to generate energy that the cells can use, and so on. And so we now know that this internal organization provides structure inside the cell. But today we're thinking about cellular movements. And let's ask which organelles are important for that. Here, I actually want to share a beautiful micrograph. You wouldn't expect David to have taken that while casually strolling through my lab one day. So he just comes up to the microscope and shoots a picture like that. Isn't that wonderful? And so inside these cells, I have to confess, these are mammalian cells. These are not fly cells, but they're nonetheless beautiful. Inside these cells, in green, you see cytoskeleton. You can see these cables in the cell. They provide mechanical functions. They provide support and the molecules that move things around. In red, we see the mitochondria, which provide the energy source for these cells. And of course, the nucleus shown in blue here provides building instructions and for making even the mirror cells themselves. And so I have to share with you, this is one of our favorite toys. This is a confocal fluorescence microscope that allows us to take these wonderful images. Well, we couldn't haul this thing here today, but we brought two fluorescence microscopes here for your enjoyment during the intermission. And my students will be helping with showing you some fluorescent larvae and embryos. Don't miss that during the intermission. But let's go back to our cells. So gastrulation involves multiple movements of cells. So what happens inside the cells that allows them to move? We can largely subdivide these events into two large classes, if I may say. One way is to build long filaments inside the cells, and they represent the main cytoskeletal elements. I can call this process sort of build and destroy. If you build a long enough filament, you can push part of the cell forward. The cytoskeletal structures are very dynamic. this video is showing you actually purified molecules that comprise cytoskeletal elements. And you can see that even by themselves, they're capable of building and then rearranging these structures. And a second way to move things inside the cell is the following. If you take these filaments that the cell can build and stabilize them in some way, then you can send other molecules along them to use as tracks for movement. And so, you know, we know our cars have motors. So cells have molecular motors in order to move things around. And in this way, they can walk along these tracks and they can pull things behind them. They can be vesicles, as the one shown here, or they can pull whole mitochondria, or they can pull other filaments relative to one another. And so this way, they can just walk along these tracks. And both types of movement require energy input. Nothing is free. And so the cells continuously provide this movement with energy. So we have seen these two types of movement, but there is one very important missing component here that allows cells to move. This is the attachment to the substrate. If you take that little amoeba cell and put it in a drop of water, it will just tumble around and will not know what to do. It needs a substrate to crawl around, like the dancers need the dance floor to attach themselves to and move around. And so do the cells need to crawl on something. And so the third class of molecules that really helps with this process is shown here. They're called integrins, and they connect the cytoskeletal elements inside the cells, such as these cables, to the extracellular molecules present in the substrate around them. And so they actually undergo very large movements by themselves. They're large at the level of these molecules, and they allow the cells to attach themselves to the substrate and crawl around. So, to summarize the first part, what have we seen so far? The cells have a really busy interior. There's a lot of stuff inside. But the various organelles, the little organs that perform specific functions, bring order to the internal environment of the cell. The movement is carried out by cytoskeleton and associated molecules, the long filaments and the molecular motors that can walk along them. And importantly, cells are mechanically coupled to the outside, so they are attached to the substrate. So this is the end of part 1.